Welcome to MRU E-Learning powered by Malla Reddy University. I am Dr. R. Kautam Kumar, Assistant Professor in the School of Agricultural Sciences, Malla Reddy University, Hyderabad. Today, I will be taking you through a very important topic in plant pathology. So, plant-virus interactions, where we discuss how viruses infect plants and how plants respond and why this subject is so crucial for the global agriculture. So, these are the contents of topic. Coming to the significance of the plant viruses are among the most serious plant pathogens globally, responsible for enormous agricultural losses every year. Because of climate change, their geographical ranges are expanding, allowing viruses to infect new crops and previously unaffected regions. Current estimate shows that plant viruses diseases cause about $30 billion US in crop process annually, which highlights their economic importance. As the global population grows, our challenge is twofold. One is to increase their productivity and reduce crop losses. So minimizing virus-induced losses is therefore essential for food security. This makes the study of plant viruses interactions one of the most crucial and critical areas in the modern plant pathology. So what is a viruses? So viruses do not think. So when we talk about viruses, we must remember that viruses are not living organisms in the traditional sense. They do not think, they do not make decisions, and they do not pursue goals in the way organisms do. They are just passive agents, completely dependent on the host cell. So a virus exists in the two phases. One is virion and one is the infected cell. The virion, which is inactive particle outside the host, and the infected cell phase where the viral genome becomes active. So plant viruses consist mainly of a nucleic acid, either DNA or RNA, and it is surrounded by a protective protein coat. Since they lack their own metabolic systems, every step of the replication, movement and protein production depends entirely on the plant cell. So, in simplest terms, a virus is an organism with two distinct phases. The virion is packaged infectious particle that travels between cells or plants, but it cannot replicate. Only after entering the plant cell does the virus switch to its active phase, where the genome is uncoated and expressed and replicated. This dual existence makes virus unique compared to other pathogens. Unlike fungi or bacteria, viruses cannot enter plants or cells on their own. They require wounds or insect feeding to gain entry. Once they enter inside, they hijack the plant's own metabolic machinery to replicate. So this is the slide explaining about the size of the viruses. So if we compare the plant pathogens, viruses are by far the smallest. A plant cell is huge in comparison, and even bacteria or molecules are much larger. This tiny size allows viruses to pass through minute wounds or to be carried efficiently by the insect vectors. Because of their invisible under light microscope, their discovery was historically challenging. So here in this picture we can see a nematode, a fungi, a bacteria and the molecules. So here the size of the viruses depends. So one exception to the size of the viruses is Pandora virus, which we can see under light microscope, which we can grow up nearly 1 micrometers and can be seen under light microscope. This exception proves the incredible diversity within viral groups. So how old are the viruses? So molecular evidence suggests that some viruses, particularly marine retroviruses, originated over 450 million years ago during the Ordovician period. So some hypotheses even propose that viruses may have existed before cellular life evolved. So this makes them some of the most ancient biological entities on the planet. Coming to the history of the plant viruses, so these are the ancient references. So the impact of the viral diseases has been recorded throughout human history. So these are the references from 700 BC describing conditions resembling rabies. Historical records highlights how viruses have been infected by both plants and animals. So in this picture we can see a human infected with a polio viruses. So, this years back, the virus has been infecting plants and those as well as animals. So, coming to the immunization history, so there is a technique known as Virolation technique which was followed in China in 11th century. So here, this technique was mainly followed to control the smallpox. So in this smallpox, where an infected pustules were taken and were injected to a healthy person. So this healthy person showed immunized to this smallpox disease. So this technique was observed by Lady Montagu, and this technique was brought about to Europe by this lady. So survivors of the smallpox protected against this disease. So this technique was observed by Edward Jenner, and he developed the vaccination. Therefore, he is called as a father of vaccination. So concept of microorganisms. The understanding of microorganisms was shaped by pioneers like Leeuwenhoek, who first observed microbes, Louis Pasteur, who discovered the spontaneous generation, and Robert Koch, who established postulates connecting microbes to diseases. So their discoveries laid the foundation for identifying virus as unique infectious agents. So how the virus were discovered? So in 17th century, the tulip color breaking disease puzzled growers. As this disease occurred, tulips has demanded more flower in the European countries. So later in 1892, Ivanovsky showed that the infectious agent causing tobacco mosaic virus disease passes through the filters that trapped the bacteria. In 1898, Besering concluded that the agent was not a bacterial but a contagious living fluid, which he named virus, meaning poison. So later, in major milestones in virology, so throughout the 20th century, important breakthroughs occurred. So Stanley crystallized the TMV in 1935, showing the viruses could behave like chemicals. So DNA was proven as a genetic material in 1944. The Hershey-Chase experiment in 1952 reinforced this. The structure of the DNA was discovered in 1953. In 1956, TMV RNA was shown to be infectious, proving that RNA can function as genetic material. So these findings transformed our understandings of molecular biology. So coming to the classification of the viruses, so mainly the viruses are classified based on the type and sequence of nucleic acid, whether it is DNA or RNA, and it is capsid symmetry. So presence of lipid envelopes, whether it is enveloped viruses, non-enveloped viruses, and virion size and dimensions. So these parameters helps virologists understand virus behavior, replication, and epidemiology. So coming to the genome types of the viruses, there are many types like single-stranded RNA, positive single-stranded RNA, negative single-stranded RNA, single-stranded DNA, or double-stranded DNA genomes. So each following different replication strategies in the host cell. So single-stranded RNAs are the most common and are capable of direct translation once inside the cell. So these are the examples of the single-stranded RNA viruses. So these are the examples of the negative single-stranded RNA viruses, and these examples of the double-stranded RNA viruses, double-stranded DNA viruses, and the single-stranded DNA viruses. And some more examples are single-stranded retroviruses, that is called as a pseudoviridae. So this is a retrovirus. So these are the electron microphotographs reveal the diversity of the plant viruses structures. So here this is a rod-shaped particles of TMV. So flexuous thread like potato virus Y and sugarcane mosaic virus. Isometric viruses like cowpea chlorotic motile virus, and bacilliform viruses like the cauliflower mosaic virus. So this is a simplified RNA virus multiplication inside the cell. So this is a typical example of RNA virus infecting the cell. So once a virus enters into the cell, the RNA genome is released, translated, and used to produce replicase enzymes. So replication occurs, new viral particles are assembled and they move from cell to cell through plasmodesmata. So systemic infection happens via the vascular system, and insects such as aphids, whiteflies, and act as a major vectors. So this entire process is rapid and tightly dependent on the host resources. So after replication, the viral genome starts translation. So during translation, the viral RNA uses the plant's own ribosomes, tRNAs, and elongation factors to synthesize proteins. So these include code proteins, movement proteins, and replicases, each essential for completing the viral life cycle. So these are the examples of the genomes of tobacco mosaic virus and genome of potato virus Y. Tobacco mosaic virus contains a simple positive single-stranded genome. A potavirus uses a polyprotein strategy where a single long protein is cleaved into multiple functional units. So studying these genomes helps us to understand how viruses optimize their limited genetic space. Coming to the virus movement, after replication, virus must move throughout the plant. So movement proteins modify plasmodesmata to allow viral RNA to pass from cell to cell. The virus then travels from the epidermis to mesophyll cells and ultimately into vascular bundles for systemic spread. The plant cytoskeleton supports this intracellular transport. So virus movement. So this is a photograph depicting the virus movement. So in this leaf, they have agro-inoculated the virus with GFP protein, that is known as green fluorescent protein. So the virus starts moving from the leaf to the plant, and this the plant through phloem and xylem, the virus move from the entire plant. So viral infections reduce the absorption of these compounds in apical leaves. They reduce the nutrient absorption in young leaves, accumulating and also cause misfolding of thylakoid membranes. As a result, pigments degrade, chlorotic patches appear, and leaves become distorted. So these symptoms reflect deep metabolic damage within the host. So what are the host functions that were used by the virus from the host? First one is to reproduce, virus use host synthesized amino acids and nucleotides, host ATPs and NTPs, host ribosomes and translation machinery. In essence, the plant provides the raw material, energy and machinery for the virus replication. So this is a plant virus interaction spectrum. So in this case, the plant survives means the virus cleared. In this case, this is called as a resistance, where the plant is survived and the virus is cleared. The exact opposite is a situation where the plant death occurs and the virus multiplication occurs. So this case is called as a susceptible. So resistance is complete removal of virus. Susceptible means complete killing of the plant. So in this between the terms, there is a term called as a tolerance. So outcomes of the plant virus interactions. So there are the three common outcomes: susceptibility, where the virus multiplies freely and cause severe symptoms; resistance, where the R gene block viral spread often through localized cell death; tolerance, where the disease persists but cause minimal harm. So these outcomes shape disease severity and crop losses. So plant viral multiplication is much more complex. So whenever a virus multiplies, plant defense responses stop the virus replication. So in order to counteract this defense mechanisms, virus also have strategies to hijack the plant machineries. So it's a two-way process where one is trying to overcome the other and the other also overcoming try to others. So this is here we can say this tug of war is running between the plant and the host. So mechanism of R gene mediated resistance. So whenever a virus enters into the plant, so the plant starts resisting towards the gene function. Whenever virus enters into the leaf, so it starts programmed cell death, that is called as a hypersensitivity. So through hypersensitivity, the plant redirects the growth of the virus and thereby restricts the growth through localized cell death. This is called as a hypersensitivity. So TMV triggers the N-necrotic resistance gene mediated hypersensitivity response. Through hypersensitivity response, there are many signals that can be upregulated, that is called as SAR, jasmonic acid, nitric oxide, salicylic acid and reactive oxygen species. So through these signals, the plant become overall active and show resistance to the plant viruses. So to counteract this R gene mediated virus resistance, so viruses also employ mutation, and also because of RNA dependent RNA polymerase lacks proofreading ability, mutations are frequent, allowing viruses to escape recognition. Some viral proteins like CMV movement protein and cauliflower mosaic P6 suppress reactive oxygen species and salicylic acid accumulation, weakening plant immunity. Coming to the very important topic, so plant possess antiviral immunity called as RNA silencing. So during infection, double-stranded RNA forms within the cells. So this, after forming double-stranded RNA, this dicer cleaves this into single-stranded RNA, so which are loaded into a RISC complex. So RISC then targets the viral RNA for degradation, reducing the virus accumulation. So this is an overall picture depicting the gene silencing. So here, whenever during the viral replication complex, double-stranded RNA is formed. So this double-stranded RNA is cleaved by a dicer protein into a short interfering RNAs. So this short interfering RNAs will move into a complex known as RISC complex, which is known as RNA inducing silencing complex, where organoid protein plays a major role and cleaves the single-stranded short interfering RNAs into single-stranded RNA. So this single-stranded RNA move to the translation machinery and blocks the gene expression. So this whole process is called as a gene silencing. So whenever gene silencing occurs into a plant or in a cell, so these signals pass from cell to cell, and from cell to cell it moves through xylem and phloem, and the whole plant becomes active through this silencing mechanism signals. So this is a picture depicting how a plant defends virus against cognate viruses by gene silencing mechanism. So in this top we can see healthy leaves, whereas in the bottom we can see the infected leaves. So when a virus infected these leaves, the plant becomes active and also it combats the virus by the process known as RNA gene silencing mechanism. After the plant passes the signals to the upper portions, the plant become active and the upper leaves show the healthy growth. So whenever a plant is suppressing the virus through gene silencing mechanism, the virus also counteract the silencing mechanism by using a silencing suppressor genes. So to overcome the silencing mechanism in plants, viruses are evolved with plant silencing suppressor genes, that is HC protein, which is called as helper component protein of poteovirus, and 2B gene of cucumovirus. So through these genes, these viruses counteract the host-induced gene silencing mechanism Thereby, the virus counteract the plants and start infecting the plants. So coming to the conclusion, to summarize, plant viruses are diverse, rapidly evolving and highly dependent on the host machinery. They cause major agricultural losses and spread efficiently through vectors and human activities. Continuous mutation and selection drive the emergence of new viral diseases worldwide. So understanding the virus biology, its genome, replication, movement and interaction with plant immunity is crucial for developing advanced disease management tools. Knowledge of RNA silencing, R gene resistance and viral evolution help us to design plant resistant varieties and innovative plant protection strategies.